Hi, Jen. Can you hear me?
Yes, I can hear you.
Hi, Jen. Do you want to try the display full play mode? Looks good. How's it going? Nice watch. Hey, Jen. John Nye's here.
It's Girl King.
I think she's like doing two Zooms at once.
Is she not talking to me? She's not talking to me.
That might be true, but I think in this instance, he's doing another Zoom.
How's life in Shanghai?
It's good. Yeah. Are you gonna be DC area
Next
first
week?
oh no.
When are you gonna be up?
I'll be there February 26th to March 2nd-ish.
And I'm gonna just miss you, but I'll see you back here.
Yeah.
She's totally doing another Zoom.
Of course.
Is she gonna join us later?
I hope so. Yeah. Or maybe she's just practicing for this talk. Yep.
So I told my wife this is the penultimate Neuro Zoom, and she said she was surprised. She said, "Why is it ending?"
Yeah, that's a good question. But I think it's good to end things, start new things.
Each thing has a season, right?
Yeah. I'm organizing an in-person meeting in March in Shanghai, 2-day all ALS 200 people. We'll see how that goes. If that's good, we can do other topics.
Mm-hmm.
Organizing meetings in China is a little bit different than in the States, just because things are all so quick. Hey, Dan Chen. Hi, can you hear us? Can you hear me? I can't hear you. I cannot hear you. Maybe your microphone's not on.
Can you hear me now?
Yeah, that's good.
Okay. So
Do you
how
want to?
about I pass the slides?
Yes, please.
So can you see the slide?
Yeah. And then can you do the play mode? Yeah.
So can you see the title?
Yeah, looks good.
Uh-huh.
How's your connection? Is it okay?
Not that good now because it says that I need to exit the Zoom to update. So
What do you have to
okay.
update?
The Zoom software.
Okay. What about
Okay now.
Okay.
So how about I try again to see whether.
Mm-hmm.
It's okay, right?
Yeah, it looks good.
Okay. Thank you. So I can stop sharing.
I've been trying for 3 years to get Young Dan to give one of these talks, but she keeps rejecting me.
Why? I cannot think of any reason.
That's what I told her. But
I see. Perhaps she's very lazy, right?
no. No.
Yeah.
Hi, it's long.
That's it.
I'm gonna tell her you said that.
I didn't say that. I gave her a hoodie. I give her anyway. It's all moot point now. Yeah, it's long. We should get John to come to Shanghai this year.
Yeah.
Yeah, I'm down. I'm totally down for that.
Chinese Society meeting.
Oh, when is that? When is that, CNS?
It's July in Zhuhai. It's
Yeah.
somewhere close to Macau.
Near Shenzhen.
Yeah, I think I've been there. When we had our institute retreat, I think we went there. Yeah. So this July?
Yeah.
Well, let me know.
Yeah, start in Shanghai. We can do something here, and then work our way to there. Zhuhai has the type of you remember that Sai Kang seafood market in Hong Kong? They have the same type of thing where you just pick out what you want and they make it for you.
Oh, I've never been there.
Oh yeah, I went to one meeting there last year.
Long. Who's organizing the meeting? Are you organizing the meeting? Who's organizing?
Not me. So there's a SFN style. So like several symposium. Also there will be a joint symposium with China, Japan, Korea. So let me find out. So if you're traveling is okay. I'm sure people will invite you over.
you don't want to leave out the U.S. Brain Initiative, do you?
Yeah, no, definitely. Yeah, we definitely want to invite a lot of friends over here. Yeah, I think it's really fine, right? So you can travel in and out, and definitely.
Just need a lot of advance notice to get clearances and things like that.
Yeah. So this will be the first year. So when CNS going to annually. So before to biannually. So this year
is triannual. Okay, yeah. So let me know. Happy to come.
I'm sure we'll try to invite you over. So I'll be going every year. So be lots like try to make a SFN style. So the Chinese neuroscience it's been growing fast. More and more new talks. So
yeah. So definitely, I mean, if you're inviting Japan or Korea, you should definitely at least invite us.
Yeah. Sure. Yeah, I was trying to get it every year, so that'd be a lot of.
Okay, great. Yeah, let me know. Look forward to that.
I'm here now.
Fantastic. Thanks, Jen.
Did I miss something? Is everything good?
Do
you have another meeting, Jen?
Yeah, I made a mistake. I double booked myself for 5 p.m. today, so I kind of took care of the other one just now. So I'm here now.
Great.
Awesome.
We're just reminiscing about our last trip to Shanghai.
Oh.
Have you met Zilong before, Jen?
Yeah, I met him before. Yes.
Okay.
In the Suzhou somewhere, yeah. It's
Yeah. Cool.
yeah,
Good to see you again.
There are several friends of mine around. So in China, they try to keep the NeuroZoom continue the style series. So I have no idea how to do this. They are trying to make several participants to host the meeting. I don't know. Maybe I just start a WeChat group to talk about it. They love it so much. They don't want the NeuroZoom to be over. So
okay. Why don't yeah. So maybe someone, they can keep the Zoom channel. Maybe someone else. Maybe Dan Chen can be the new director of it, and she can.
Yeah.
Hey, Dan Chen. Hey, everyone.
Hey.
Yeah, sorry, I missed so many meetings.
Yeah, we just find a time to compatible with the offline meeting. So if you are busy right now.
Yeah,
so,
it's crazy.
Let's talk about it.
Mm-hmm. Yeah, definitely. Yeah, the teaching is really crazy nowadays. Yeah.
Oh, okay.
How much do you teach?
So I teach 3 courses every year.
What?
Yeah. Yeah, it's a little bit too much now. Yeah, and most of them like don't have a TA, so it's a little bit crazy.
I teach one class for 3 weeks, 15 students, and I have 2 TAs.
Wow.
Yeah, it's a private university. You guys barely work.
Yeah. So it's kind of.
Hey, Da Wang's here. Hey, Yimin.
Yeah.
Da Wang.
Yeah, Da Wang.
Hi, Jane. Long time no see.
Yeah, it's been a while.
Didn't see what the ski trip, ski meeting, you mean?
Yeah. I missed that ski meeting.
Yeah.
It was like yesterday.
Yeah, it's really fun. Yeah. Next year they are going back to Colorado.
Okay.
Yeah, so you should definitely come.
Aaron, we should apply for a symposium. Oh, actually, last time Aaron was supposed to be in our symposium in that meeting. When I saw you, he actually couldn't make it.
But I sent someone else who did a great job.
She was a great speaker.
Yeah, I trained. All right, maybe we'll start. Zong, is that good?
Yeah. Yes,
okay. All right. Hi, everyone. Welcome to the second to last NeuroZoom, at least ostensibly in this iteration of it. Looking forward to awesome talks. Next week will be the grand finale of NeuroZoom. So please tune in to hear the latest from me and Zilong. We'll have thoughts about NeuroZoom. We'll talk about some stories from our lab. I didn't plan this orange background, orange this, and orange buttons. But anyway, please tune in. And it's been great meeting with all of you for almost 3 years. We'll have more to say about that next week. But for now, let's get started with Zilong can introduce our first speaker, Dan Chen.
Sure. Okay, it's my great pleasure to introduce Dan Chen Liu, who is the PI in ION. So I'm very happy because when I first come to ION, Dan Chen is just the first year graduate student. Now he's a principal investigator and has done a fantastic job at ION in her new lab. So Dan Chen has got her bachelor's degree in USTC. So after college, she went to ION. So inspired by Professor Mo Ming-Fu, she joined a group, Mo Ming's group, and worked closely with the postdoc Yang, who is now a professor in Shanghai Tech. So has started an ambitious project to analyze the memory trace in the from amygdala to auditory cortex, which work published in the National Neuroscience, combining many cutting-edge technology, including visual imaging, optogenetics, and many beautiful works. So after PhD, Dan Chen went to Yang Dan's lab in Berkeley, where she started work with in mechanism with memory. So after a lot of beautiful work, I don't have need to see. So she started her own lab in ION a few years ago. So quite a short time period, she published the first work in her own lab, where she discovered a beautiful mechanism underlying the rapid eye movement REM sleep and cortical regulation of two-stage REM sleep, which is beautiful, quite just amazing. So I believe today Dan Chen will talk about the new work in her new lab. Okay, Dan Chen, take it away.
Okay, thanks for the nice introduction. So okay, can you see the first?
Sure, you're gonna go to the slideshow. Oh. Okay, great. You're muted, Dan Chen. You're muted. The screen is great. Yeah.
All right. Just to update the software or something. So can you see the screen now? No.
Not yet. Yeah.
Sorry for this. So it's okay now?
We can see it, but you not start a slideshow mode.
I started.
Yeah, it works.
So something always happened. So once I share the full screen, then
it looks fine now.
Error comes.
Yeah, now it's working. Take it away.
Okay, so how about I just show it here?
No, it works. A moment ago, it works.
It was fine before.
Yeah.
Okay. So it's okay now?
Yeah, works.
Okay.
Go ahead.
Okay. Sorry. So today I will talk about the role of cortex in rapid eye movement sleep. Rapid eye movement sleep is a very specific sleep stage with a very wake-like activated EEG and the paralysis of EMG. It's also called a brain-on, body-off sleep state, paradoxical sleep state, associated with rapid eye movement and vivid dreaming. So every night we progress through several stages of non-REM sleep to REM sleep for 4 to 5 cycles. And when we wake up from the REM sleep, we usually could remember the content of our vivid dreaming. However, we still need know little about the mechanism and function of rapid eye movement sleep and its associated dreaming. So in the last decade, people have identified a bunch of subcortical circuits regulating non-REM sleep, but much less about the REM sleep. And cortical activation has been considered to be associated with the dreaming content. As shown in human studies, in REM sleep with dreaming experience, the posterior and medial part of the cortex got much higher activation, and activity of the visual cortices could predict the content of visual imagery during dreaming. However, the role of cortex in REM sleep regulation has never been examined before. To characterize the global cortical dynamics during REM sleep, we performed cortex-wide calcium imaging on head-fixed Taiwan mice with the skull replaced with a chronic cranial glass window, together with EEG, EMG recording and facial behavior recording. Here shows some example calcium imaging frames. The cortex is indeed highly active during REM sleep and inactive during non-REM sleep, but the activation pattern during REM sleep is clearly different from that of the wake state. So with the posterior and medial part more active and some somatosensory areas more silent. So we performed the spatial ICA analysis and identified several functional modules corresponding to several anatomical structures, such as the retrosplenial cortex, RC, parietal cortex, primary sensory cortex, etc. We averaged the calcium signal within each module and aligned with EEG, EMG, and brain state. We can see here that RC visual cortex got activated in every REM episode, but activation of the RSC is distinctly higher than all other cortical areas during REM sleep. And although almost all brain areas are more active in REM sleep than in wake, the difference is much higher for the RSC. We next performed the granular causality to examine the activation sequence of the cortical modules. As shown here, we can identify significant causalities from RSC to many other cortical areas, but none from other areas to the RSC during REM sleep, suggesting an initiating role of the RSC in cortical dynamics. To further characterize the spatial temporal patterns of the cortical waves, we performed the seq-NMF analysis to extract the repeated calcium events. Across mice and sessions, we could identify 7 common motifs, with 3 of them selectively active during REM. More interestingly, in all these 3 motifs, the activities all initiates at the RSC, either local activation or propagating from the RSC to visual cortex or anteriorly to the ACC. So here, all these wide field imaging studies show that the RSC is selectively activated and initiates the patterned cortical activity waves during REM sleep. To take a closer look at RSC activation, we performed 2-photon imaging on layer 2-3 or layer 5 RSC neurons. During REM sleep, we could see that most layer 2-3 neurons got activated, and on average, they are selectively REM active. Here, each line represents the average activity of all neurons recorded in one single session. By contrast, the layer 5 neurons were mostly silent during REM sleep. We also recorded from the M2 layer 2-3 neurons. Here shows the REM modulation of each individual neurons we recorded. We can see that most RSC layer 2-3 neurons are REM active, and the layer 5 neurons are REM inactive. But the M2 layer 2-3 neurons were not modulated much by the REM sleep. So here we can conclude that the cortical activation during REM sleep is both region and layer specific. So back to the RSC layer 2 activity, we also observed the divergent temporal patterns. So with some neurons fire very early at immediately after REM onset, but others activated much later. We measured the pairwise correlation coefficient and indeed observed two distinct clusters. And these two subtypes of neurons, they showed largely non-overlapping temporal profiles, with the type 1 neurons mostly activated in the first 20 to 30 seconds, and the type 2 neurons more active at the late stage. We thus wondered what could happen at this transition time point. So when we inspected the mouse facial video, we strikingly also found that this heterogeneity. In the first few of seconds, we just look at a few seconds before, they are totally silent. There is no movement at all. But later, they are enriched movement, including the eyes, the eye whisker, and the nose, and some jaw muscles. So they are this movement interleaved with each other, representing a sustained activation state at the late stage of REM. Using a facial expression analysis, we obtained the outlines of the mouse face and calculated the pairwise CCs of each video frames, followed by hierarchy clustering. So here, the cyan box represents the frames within a REM episode. We can see there are two main clusters within one REM episode, with the first one similar to the long-REM sleep, and the second one showed enriched facial movement, and the movement pattern is distinct from that of the wake state. So we thus define the first cluster as quiet REM, QREM, and the later as active REM, AREM. So these two substages is also associated with differential EEG oscillations, especially in the theta band, with the QREM showed a much higher power in the low theta from 6 to 8, and AREM showed higher power in high theta, 8 to 10. So we calculated the ratio of low theta to high theta, and this ratio is significantly much higher in the QREM. So here, after the transition, we can see there is a peak in the QREM and a drop during the AREM. And eye movement and facial movements are much higher during the AREM than QREM. Moreover, the heart rate variability, respiration rate, and hemodynamics were all significantly different between the two substages, with the AREM showed a much higher sympathetic tone with a decreased HRV, higher respiration rate. So next, we want to say whether these two substages could be identified in the natural REM sleep in freely moving state. So we recorded the facial EMG together with the EEG and the neck muscles. And indeed, we can see clear higher facial EMG tones at the later stage of the REM sleep, correlated with the drop of low to high theta ratio. So here is a clearer demonstration. So while QREM and AREM belong to the REM cluster in the theta versus neck muscle tone plot, they fell in distinct clusters when we plot the low to high theta versus the facial EMG. So this demonstrates that the two substages we observed in the head fixed, we found in the head fixed condition could be applied to the natural REM sleep. So back to the RSC layer 2-3 activity, we found that the transition of the type 1 and type 2 activation matched well with the QREM to AREM transition. And also the type 1 and type 2 neurons either showed a negative or positive CC with the AREM, the later stage of the REM sleep. By contrast, they both showed a very low CCs with eye movement unknown, indicating that their activation are not like time locked to the eye movement events. So we next trained the SVM classifier to see whether the population activity could encode the two REM substages. We can see here that once we use the more than 60 neurons, so the prediction accuracy exceeded 90%, and the accuracy is much higher than when we decode eye movements. Moreover, So the RSC layer 5 neurons, they were more silent during the later stage of ARAM, and the QRAM to ARAM transition aligned with the activation peak in the wide field imaging data. So all this suggests that RSC contained enriched information to encode the 2 RAM substages we identified. So here, to summarize this part, we found that the REM sleep can be divided into 2 distinct substages, a quiet QREM stage and an active AREM stage. So they can be characterized by the differential EEG, theta oscillation, facial movements, and autonomic profiles. Moreover, the RSC layer 2-3 neuronal activities could encode the 2 REM substages. To further examine whether the RSC is essential for the cortical dynamics or the REM substage regulation, we performed the optogenetic inactivation experiment. So we injected the virus expressing NPHR into the cell-angiocamp mice, so we can achieve concurrent optogenetics and wide field imaging. So once we detected the REM sleep, we deliver yellow light into the RSC. So due to the relative low efficiency of inactivation efficiency of NPHR, we could see that RSC activation was not completely abolished, but still significantly reduced during REM sleep. And RSC inactivation instead caused a general increase in other cortical areas. So here shows the differential map, inactivation versus minus the control group. So here means a general increase in other cortical areas. And compared to the control group, the occurrence of motifs with RSC as an initiation site all dramatically reduced. So we further tested the RSC inactivation at the behavioral level. So here we use the virus GTACR with a higher inactivation efficiency. We deliver the blue light throughout the REM episode. We can see that in control group, most REM episodes is composed of 2 sequential QREM and AREM. However, in inactivation group, most several episodes contain only the QREM, but with in the episodes with AREM, the duration is much shorter. So here in this summary, we can see that RSC inactivation during REM sleep largely decreased shortened the AREM duration, prolonged the QREM duration, leading to a decrease in the total duration of REM episode. So this effect was largely due to the inhibition of the QREM to AREM transition, with the maintenance of QREM largely enhanced. So during the recording session, the frequency of REM episode largely increased, suggesting an enhanced REM pressure once we interrupted the REM sleep. By setting a delay after REM onset, we also achieved the AREM-specific RSC inactivation, and we can see that the AREM maintenance is also largely affected. But if we inactivate the RSC during REM sleep, either wake generation or REM generation is not ordered. So this means that the RSC plays a selective role in the REM maintenance, but not other brain state transition. Finally, we also inactivate other cortical areas such as the M2 and ACC, and found that they have no effect on the REM sleep duration or frequency. So this part of the optogenetic inactivation demonstrated that RSC is important for the cortical dynamics during REM sleep, because inactivation of RSC largely alters the cortical dynamics and inhibits the QREM to AREM transition and AREM maintenance. So to summarize, in this work, we found that the REM sleep in mice can be divided into 2 distinct substages, and RSC plays an important role in regulating REM sleep progression. So in the future, it will be important to examine whether such 2 similar substages also exist in human sleep, perhaps R1 and R2, and whether they play differential roles, because currently we still, the role of REM sleep is very controversial in emotional processing or memory processing. People got very contradicted results. So perhaps it's because like the heterogeneity of the REM sleep structure itself. So at last, I want to thank Dong Fan. So a very talented PhD student who is still in the 3rd year, and she made most of the these discoveries. And also thank Jiaqi, Ming, and Yihui. They contributed a lot to this work. Thank other lab members, and hope all of us can sleep well and have good dreams. And happy Valentine's Day. Thank you.
Thank you, Dan Chen, for a beautiful talk. It's amazing.
Thank you.
Congratulations for your discovery. Now, open questions.
So Dan Chen is here. It's a great talk. Really
Thank
exciting.
you. Thank you.
Yeah. So I have a very naive question, or maybe it's more complicated than I thought. So I found like in your second part is a QRAM, right? So you have more movement, but your layer 5 is silenced.
Yes.
How do I understand that? Or maybe even more complicated for the REM sleep, or whole body is no longer moving, but with the facial movement, how do they disassociate?
Yeah, that's a very good question. So first, on the neuronal activity level, we do see like that in the layer 5 neuron, at least in the areas, we didn't check the layer 5 neurons. So in the RSC, the layer 5 neurons is really silent during the REM sleep, but the layer 2, 3 and activated. So we are thinking that perhaps this is like although we like the consciousness in REM sleep, we still feel something goes on during the dreaming, but it's distinct from that of the wake, right? And also because most of the layer 5 neurons, they are PT neurons, right? So for the body control, for the body muscle control, the PT neurons perhaps is the most important ones, right? So I think the inactive of the layer 5 neurons could be related to the muscle atonia or the paralysis of the peripheral system.
Yeah, but for the facial muscle, are they on the?
Yeah, for the facial muscle, we are still do some tracing work to see like how because for the facial muscle, their control, the motor nuclei in the brainstem is much anterior than the skeleton muscles, skeletal like peripheral motor system. So we are thinking like the Atolian system, the gating system didn't work on this facial muscle control mechanisms. So that's why we can still observe the enriched facial movements. So because previously we already all know that the rapid eye movement sleep, right, is because the occurrence of rapid eye movement. So here we also identify the other enriched facial movements.
I see. Yeah. Oh, that's interesting. Thank you.
Thank you.
Okay, and more questions. Okay.
Hi, Long. Can I have a question? Sorry.
Please go ahead.
Yeah. This is Yunfeng from the Chinese Academic Sciences, the Institute of Zoology. So it's a pro story. So I have also a naive question for that. So are there any other physical role of the RSC for in the animal models? So
the RSC is
besides besides for the sleep cycle, yeah.
Yeah, the RSC is very important for the spatial information encoding. So they received a very so interacted with the hippocampus, the speculum and the CA1, and there are many neurons encode the landmarks and the head direction and other spatial information. And also the RSC is also important for the social interaction. I think Zeno has a very interesting work on this. They show that in the autism, once the RSC goes around, that the social interaction also.
That's weak. That's weak. Okay.
Yeah. So the RSC is the most two important roles of RSC is and also memory. So the memory processing, I think it's also because of the interaction with the hippocampus. And people think that RSC is like a bridging the hippocampus with other cortical areas. So we think that there are many other, so because when we observe the dynamics of the RSC layer 2-3 activation, we found that during each REM episode, like the activation is very dynamic. So I think perhaps they encode some complex information to process during REM sleep, but we are still have something going on that.
Sure. Another question is, I saw that you have like do some loss function so that you inactivation of the those neurons in like in the RC. So what's the other way around? So when you like inactivate those neurons, what will happen? So do I mean the mind change from the like to the IM to the non-IM like that, or what will happen?
Yeah, we tried that before, but
Huh?
but unfortunately, the activation of the RSC, the cortical neuron, the synchronized activation of the cortical neuron sometimes cause the seizure-like phenomena.
Ah.
Yeah, so we can see like the propagating calcium waves, like very much like the seizure, and from the behavioral level, we can see that the mice got abnormal. So perhaps because of the synchronized activation pattern, right? So we didn't get the results of the RSC activation in the behavioral, so in regulating REM substages.
Okay, thank you. Also, thank you, Flo. Thank you.
Thank you.
Okay, Jimmy had an important question in the chat bar. Do you want to ask yourself?
Okay. So given the potential role of the RSC in spatial information, do you think that this finding is specific to rodent REM sleep in which viscous spatial mapping occurs, whether occurring in humans? What do you think of the role of RSC might be? Yeah, that's a very good point, because the mice use the spatial information much more than us humans, right? So we have two things, like we plan to do two kind of things to further strengthen this point. So first, we want to like increase the other like visual experience or other experience to see whether the experience, because the enhanced visual experience selectively enhance the activity in the visual cortex. So that's a very indirect evidence actually. So back to the human REM sleep, what do you think the role of the RSC might be? So we check some EEG data in the human REM sleep, but currently we didn't like take some useful information, but we still think that RSC could be important, because from the anatomical level, we did some tracing of the RSC and found that the RSC received a very dense inputs from the pointing area. So the pointing area is the classical REM center, REM sleep generating center. So we think that the RSC could play a role like in mediating because the pointing waves is very important for the visual imagery during dreaming. So people think like the pointing waves from the pointing to.
Hello? Dan Chen is frozen. Is it? Okay.
Okay, so we think, so back where are we? Well, so I'm talking about like the RSC could be also play an important role in mediating like the pointing signals and propagating to other cortical areas. So we think that this could still be true in the human, and also during the human dreaming with human REM sleep with dreaming experience. So people have, using the like EEG studies, people have found that the medial and posterior part of the cortex is highly active, including the RSC region.
Okay, thanks, Dan Chen. I have one last question for my privilege of being host. So I have a brutal hypothesis of what RSA might do. So let's say you find out that in the REM sleep, lucid dream has associated with the activation of the RSA. So we and others find out that the RSA is quickly activated during social interaction behaviors. So that's a tight associated with some everyday scene that animal might imagine, including human, right? In the scenario where there's normal social interaction with the dreaming and lucid dreams. Yeah, I think RSA might play an important role in replay or replay the normally everyday scene in the in the social behavior or in the dreaming scenario. All right, that's my comments. You do need but let's if you maybe have some way to find out in the future.
Thank you. Thanks, No. So I think the most important thing is to identify the cell identities in the RC, right? Either the spatial encoding or the social interaction. So once we identify those neurons, like sorting out those subtypes and test that their activity during RMC.
Great. Thanks, Dan Chen. So
thanks, No and Aaron.
Yeah, now a second talk for Jin's talk. Aaron.
Hi. Okay, great. Thanks, Dan Chen. Awesome talk. And now it's a great privilege and pleasure to introduce Dr. Jin-Young Lee. Jin is my colleague at Stanford. She's my best friend at Stanford, so it's a special treat that she can give a talk here. Jin received her bachelor's degree at Seoul National University in electrical engineering. She then came to Stanford, where she did a master's degree and PhD, also in electrical engineering. She then did a postdoc at Stanford, and then after a brief time at UCLA as an assistant professor, she was recruited back to Stanford, where she's currently an associate professor in the departments of neurology, bioengineering, neurosurgery, and electrical engineering. Jin is the quintessential interdisciplinary scientist. Her goal is to define and manipulate brain circuits. Her research brings together widely diverse fields ranging from signal processing, imaging, genetics, neuroscience. She builds hardware, literally builds hardware. For example, she can make MR compatible optrodes to make electrophysiological recordings while an animal is awake and behaving in an MRI machine. She literally writes software, brings in her knowledge of the signal processing field. She develops compressed sensing algorithms for high spatial resolution fMRI. She doesn't just use these techniques, but because of her deep engineering knowledge, she knows the idiosyncrasies of these methods and harnesses these to her advantage. She then brings these innovative technologies to bear on key questions in fundamental neuroscience and disease, including, for example, studying how stem cell-derived dopaminergic neuron grafts can integrate into brain circuits for Parkinson's therapies. She develops methods to improve the efficacy of deep brain stimulation, so on. She had this amazing paper in Nature where she invented a technology called optogenetic fMRI, or OFMRI, and that has allowed her in the field to solve many big questions. For example, fMRI is built on the assumption that brain activity is correlated with blood oxygen level dependent or bold signals, but this had never been formally tested. And she used this technique to show that this was positively correlated with neuronal activity, both at the site of stimulus, but then importantly, she can stimulate neurons and then read out the effects in distal parts of the brain. This technique really allows, compared to conventional approaches like fMRI, allows global analysis of brain activity. Jin's won many awards. She's been a Sloan Foundation Fellow. She won an NSF Career Award. She won a NIH Director's New Innovator Award. She won a NIH Director's Pioneer Award, and so on. So looking forward to hearing the latest, Jin. Thank you.
Can you hear me?
Yes.
Oh, thank you, Aaron. That was a very long and flattering introduction. Thank you. Today, I just wanted to give you guys like a brief overview of some of our recent work. So it's titled Solving Brain Circuit Function and Dysfunction with Computational Modeling and Optogenetic fMRI. As Aaron has introduced, I am actually fully trained in electrical engineering. And then I actually became a professor in electrical engineering and then decided to change fields. And so now I am running a neuroscience lab. This is my lab at Stanford in the Stanford BioX, which is where I'm sitting in right now. And I wanted to also give a little advertisement about the new system that we now have at Stanford. It took me about 10 years to house this system on Stanford campus. It's a neuroscience preclinical imaging lab, which is also open to others in other campuses and industry as well. And so we had an inaugural symposium for that in last year, April, and we're we'll have another one in May of this year. So to just simply give you an overview of what my goal is in my lab, brain health care, brain disorder treatments in general, the ultimate goals are to restore brain function. And so it is obviously very important to understand how the brain circuit elements communicate in order to restore this function. And so my lab's general goal has been to take a certain behavioral unit and reconstruct the brain circuit communication algorithms underlying the behavioral units. And in general, having tools like optogenetics makes it easier to identify certain behavioral units. And so here, this particular video is D1 stimulation with optogenetics. And on the right is actually a circuit that we reconstructed from it. And so just to give you a brief example, like I said, D1, D2 MSN stimulation is one of the easier and also more obvious examples of how we use optogenetics in combination with fMRI. And so by selectively stimulating them with fMRI, we can elicit very specific behaviors, which then we do optogenetic fMRI with in order to understand the underlying circuitry for the behavior. And so this is an example of the fMRI. We can then, like this fMRI shows different activation patterns. D1 and D2 obviously show very distinct color, which indicates different responses to stimulation at exactly the same location, which was the striatum, dorsomedial striatum in this case. And since this provides 4D images of the whole brain, we can segment out different hindquarters. This is D1 stim example, D2 stim example. We can then also go to regions of interest and do electrophysiology. And for the most part, we confirm, actually for all cases, we confirm that the fMRI activation signal closely matches the underlying stimulation, electrophysiology pattern. And our goal was to construct the algorithms. And so we went and did the reconstruction of the algorithm underlying these modulations. And from the fMRI, we were able to get these interaction maps. And so the direct pathway, as you can see, has very well-known activity associated with direct pathway, and indirect pathway the same. And these are from single measurements that were all simultaneously made, which allows us to get an interaction map. This is not a connectivity, this is interaction map. And once we had this interaction map in place and had a global view of it, we could also model things at the single cell spiking level across the whole brain. And so this is an example where we can then take the D1 MSN stimulation. These are actually electrophysiology measured from the regions of interest identified with fMRI. And then these are the simulated firing rates. And so we can now simulate things at the single cell spiking level. And so this is just statistics showing that the firing rate matches in the model and the experiment. This is for D2, and you can see that, yes, indeed, we can start to model this. And by being able to model brain function, we can now do things like select an optimal therapeutic target for brain disorders in a matter of minutes because they're simulations. And so this is an example stimulation of D1 MSN stimulation, and then trying to inhibit in STN or cortex, GPI, and GPE. And you can see these are the inhibition site. This is the stimulation site, and you can see the actual brain activity simulated in all these different locations: STN, cortex, GPI, GPE, thalamus. And in short, you can see that in thalamus, the activity is inhibited only with GPI inhibition. And so this particular example, to inhibit these activities induced by D1 MSN stimulation, GPI is ideal based on our simulations. And we can actually see, without just doing our own experiments, here, this is an example of a paper from another group that shows that in this kind of model, the GPI is indeed the ideal target. And we could do this. I'm sure this experiment took a long time to do, but now we can model it and then make predictions, which seem to match the experimental outcome from others. And so this type of approach, we can now do cell type specific whole brain function imaging. We can quantitatively parameterize the whole brain scale interaction. We can now model them at single cell spiking level and just design and predict optimal neuromodulation therapy targets and parameters. And that is like one very important branch of what we do. And so we use fMRI, then many other techniques combined to model brain function. And since our interest is generally around neurological and psychiatric disorders, one other very important aspect of disorders is pathology. And so this is actually a collaboration with Aaron's lab, where one thing we wanted to do on top of understanding function, since functional restoration is the goal, the other aspect that we wanted to do was to understand what the pathology relationship to what does pathology do and how do they relate to function. And so this is an example of where we injected the alpha-synuclein fibrils into the striatum. And in this case, since this is not a in vivo imaging technique, we sacrificed the animals at different stages, 2 weeks to 18 months. So this was a very long experiment. Then we used brain clearing, in particular the iDisco method. And then we did light sheet microscopy, and you can see images that we collected. Importantly, we took this and developed all the registration and segmentation tools using machine learning and et cetera. And then so we now have a fully automated platform to be able to analyze this. And here we injected and imaged at these different time points. Like we mentioned, previous publications showed that there's spreading increases up to about 5-6 months, but we imaged through 18 months. And what we're seeing in general is that on the ipsilateral hemisphere and contralateral hemisphere, if you just take the whole thing, you can clearly see the pathology increasing and then decreasing. And also another important observation that we made was that the aggregation size changes over time. And so at different point in time, the distribution of the pathology size changes. And so, like I said, these were all fully quantified in a automated pipeline. And so here you can take these segmentation and do all kinds of different quantification. B here is showing the changes at different pairs, 0.5 to 4 months injection, 4 to 8 months injection. So different time points. Green is increase, red is decrease. And so the pathology increases and then decreases at different times. It changes its course. And then this is when you look at the density, this is when you look at the size. And so there's different aspects of all of this that we can quantify. If you want to look at a summary based on the segmentation of regions, this is another thing that we can look at. And based on the fact that they spread and also decay, and there's obviously aggregation mechanisms associated with it. And so we then use computational models to be able to fully parameterize both all of spreading, aggregation, and decay. And we see that, so for example, for the modeling, we divided the aggregation into 7 different size bins, which are all the parameters are optimizable. So we actually globally optimize all the parameters. And when we did that, we get a very nice fit of the actual pathology measured and our model. And so for different size bins, they match pathology, etc. And so with this computational model, we can start to parameterize how these things behave. And so this is an example of visualization of spreading estimates. And you can see we started with the injection in the striatum, and you can see that dominant spreading happens from different places over time. It starts in the striatum, and then now it goes to the contralateral site, and then it spreads from there. And then the most dominant one after that is from cortex. And so these you can parameterize and visualize, as you can see here. And also, since we have fully parameterized the different steps, we can start to map also genes that are associated with each of the parameters. And so this is an example of using the database of gene expression and mapping it to different parts of the model to see how the different gene weighting impacts the spreading and degeneration, for example. And so this is a degeneration axis, and then this is a spreading axis. And you can see these are the different genes known to be associated with Parkinson's disease. And for example, LAMP3 ranks high in terms of its association with degeneration. LRRK2 is ranking high in terms of the spreading, which makes total sense knowing what we know about these different genes. And here, this is an example of the spreading related parameters. So here, the highly ranked ones, the neuronal components, neuronal genes associated with neurons seem to have more association with the spreading parameter, while genes associated with degeneration is more of genes associated with oligodendrocytes and astrocytes, for example, which also makes sense. And so this is starting to give us a platform where we can make hypotheses and then test their roles in different parts of this spreading and degeneration mechanism. And so this part of the talk, I showed you that whole brain distribution and size, these can all be quantified, and we can do statistical analysis. We can then also, just like we model the function, we can now model the pathology dynamics. And then this we can visualize in different ways and also relate to things like genes associated with these mechanisms of spread and degeneration. And so this similar type of this study was actually done in order to also relate it back to function. And so in the same injection model, one of the things that we can do is see where these things spread, and we can even model it. And so just to show you some preliminary outcome of this is that we can do optogenetic fMRI to see what type of stimulation does in terms of its impact on function. And then we can also see like what it does on the pathology change. And so on the left is the neuromodulation impact on function. And on the right is the neuromodulation impact on the pathology change. And you can actually see a striking similarity where if you fully quantify it, you can see that the increase in aggregate count is related to decrease in function, and the decrease in aggregate count is related to increase in function. And so the function and pathology chain globally has one-to-one correspondence, which is something we can now start to quantify, which shows that function and pathology have very strong relationship, which has significant implications for therapeutics design. And so what I've shown here is that OFMRI measured brain function changes predict changes in aggregated protein across the brain. And using a similar set of technology, this is work in collaboration with Adriano Agusi's lab. And we also evaluated how different drugs impact pathology. And so drug is, of course, an important intervention that we want to understand in terms of starting to as a way to modulate these pathology. And so a novel brain clearing method, in this case, a different one from the iDisco method we used earlier that was developed in Adriano Agusi's lab. And this technique actually allows for faster imaging, which allowed us to process a whole lot of brain. And with this, we also showed that they have same staining capability. And then here we had a set of different age group and therapeutic application. 3 different drugs at 2 different age groups were applied 5 and 14 months. And then we quantified different things like plaque count, size, and maturity. And we used the similar pipeline to quantify things. And in this regional map, density, size, maturity, different regions for 3 different drugs. At this particular quantification level, like anti-A beta hardly does anything here, but NB360 and LIN5044, which are two different kinds of drugs. You can see very clearly that the density, in particular, density and size, has huge decrease in younger age, 5 months, but not at an older age. And in case of the other drug, the maturity and size has dramatic changes at older ages, but not at younger ages. And so this is a spatial map instead of the bar plots I showed earlier. And so here, these are 5 months, 14 months, younger age has this NB360 had huge impact on the decrease of density, and also it had different changes in maturity at different time points, different locations, and there was not very much change in the size in the young cohort, but LIN5044 had relatively no change. And at 14 months, this other drug, NB360, had no impact, but LIN5044 had a dramatic change in maturity distribution and also size, which shows that different drugs, even though this kind of evaluation has never occurred in clinical trials before, different drugs have different impact in its in different ages, as well as different aspects of the pathology, which should be an important factor to consider in drug design. And so this again shows the difference in the distribution. In summary, these clearing methods was developed for larger quantity evaluation. We can automatically calculate all these different parameters, and we conclude that region and age-specific plaque counts are important. And so overall, fMRI has the ability to image and allow us to computationally model now at the single cell spiking level, make predictions, and tissue clearing methods were combined with various computational methods so that now we can quantify them to model its mechanism of spread and decay, as well as now look at ways that we can intervene with neuromodulation, and also we can then quantify different impacts of things like drug therapy. And so with that, I'd like to thank various people in the lab who contributed to the study, Aaron's lab, Ron Melgi, and Adriano Agusi's lab. Thank you very much.
Thanks, Jen. Awesome talk. We're open for questions. All right, maybe I'll start with one. Do you see any examples maybe for the synuclein project or others where there might be anti-correlations with pathology in one brain region and activity somewhere else, sort of activity that changes sort of in an unexpected way?
Oh, that is exactly what we see. It's anti-correlated. Increased activity leads to decrease in pathology, and decreased activity leads to increase in pathology. In that example I showed you, we still have to investigate exactly how that should be interpreted and used for therapeutic design. But one thing's for sure, like brain activity changes pathology very conclusively. And so that should definitely be considered for therapeutics design.
I mean, are there examples sort of what the opposite of that? Like in unexpected brain regions? No.
Not so far.
Other questions?
0 questions? Maybe I made it really, really clear.
We're still processing all of the new images and technologies. And then what do you think about the prospects for using this knowledge about correlations with activity and pathology and to sort of change the trajectory of the pathology?
that's exactly what I think we want to do. Actively guide the therapy, actively use neuromodulation as a way to change pathology. At the same time, I think it also has implication in a sense that restoring function in a way would, in many cases, restore pathological changes that we don't want. And so, you know, there is often discussion about, from like more systems neuroscientists, oh, we want to understand all this function, but what does function have anything to do with pathology? But there's very close relationship that we can conclusively see. And so there are many ways to go from broken to restored, but in general, it means that function plays a very critical role back and forth with pathology.
Got it.
So Jen, I have a question. So this is a really cool technique, but my general question, I guess, not just for you, but for the field, is that by the time you're seeing these aggregate pathologies, you know, it seems to me that it may be kind of too late. So are there models where you can actually get insights before there's any any overt pathology like fibrils or tangles or accumulations? Because it just seems to me that by the time you're to the point where you actually see the frank pathology, you're just not going to be able to reverse the pathology. And the second part is the extent to which, you know, for example, the genetic or transgenic mouse models for AD, I think, are really targeting very late stage events and not getting at the initiating events. So how might you apply this technique to kind of getting around what I think is this quagmire?
That's a very good question and a very complex question. So one of the reasons why we did this particular pathology study is to understand its mechanism. So now we want to add various points in terms of getting at earlier information and also relating it back to function. And so this is kind of a way for us to understand some of the things and how it interacts with each other. I wouldn't necessarily say that our therapeutic goal is to intervene at a very late stage where pathology is like all over the place. At the same time, one thing that this study taught me is that even with a whole ton of alpha-synuclein pathology, the animal is mostly fine. And so there is something more than an alpha-synuclein pathology that is critical to the disease, for example. So this is an interactive learning that we're doing here, and we formed a lot of new hypotheses around it, and we're also trying to integrate all the information together to make it more interpretable. I hope that somewhat answered your question.
So a lot of people now are using to great effect deep brain stimulation as a treatment for Parkinson's disease. And you know, I wonder if like you have access to some autopsy materials or things like that to maybe test some of these hypotheses, maybe if it increases pathology in certain regions that you might have predicted.
That's a very reasonable thing to do. And I think that would be great for us or someone else to test, see if that's really the case. And I would strongly argue that that would have changed things. And so, like, just like our case of computational modeling relating different genes back, it gives us a way to make these hypotheses. And one you made just now is a really good one where we could start to go look at that because of these experiments.
There's a question in the chat. Do you think there's a correlation between neuroplasticity as we learn, along with pathology and activity, that you'd like to please comment, share thoughts on?
Do you think there's a correlation between neuroplasticity as we look? I'm not exactly sure I understand the question. Yeah, I mean, absolutely. I think there is neuroplasticity involved in this, and we're finding more increased evidence along that. And just like you asked that question, looking at our data, like we are forming these hypotheses and testing them. There's also a second question. Can we do more advanced modeling? yes, we're actually exploring a lot of different models, and the ones we've made already is making predictions. But then again, we're also looking at other kinds of models to further integrate parameters that are of interest. And so it's a process of us adding different parameters that we think are important for disease, which is my primary interest.
All right. Thanks so much, Jen. Really exciting and great to see everyone for this second to last Neuro Zoom. Next week will be the last one.
Can I ask why this is the second to last? I didn't know it was the second to last.
It's
complicated.
These are good questions. Yeah. But we couldn't stop without you. And now that we have you, we could stop.
Jen, you missed like 358 great talks, but you can find them on YouTube.
Okay, I'll watch all of them to commemorate the ending of NeuroZoom next time.
Start now. You might make it till next week.
Do you know about the NeuroZoom TikTok channel, Jen? You can subscribe to that.
And you promised a hoodie for NeuroZoom by giving this talk. Aaron's
Yes.
saying that if I don't give the talk, I'll never have the hoodie. So now, okay.
Yes,
okay.
I have the hoodie for you. Extra small. It's ready to go.
Not extra small.
Right.
All right, everyone.
All right.
Great.
Thank you. Bye-bye.
Yeah,
Bye.
great
See
talk.
everyone next week.
All right, see you next week.
Bye.
Next week.
Bye.
Bye.